Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with ShahTareen Swati. And today I'm here with the science art of the day, which is created by the Zainab Arshad. She is pre-medical student at Muslim Education System, and she created this glycolysis in a very amazing way. She used I'm showing you here the glitter foamic sheet for the carbon and the phosphate and ATP molecule and NADH molecule, and use different colors to create different molecules or compounds at different steps of glycolysis. You can see here phosphate in different color, carbons, different glucose and fructose are in different colors. Very amazing. Now, what is glycolysis? Actually, glycolysis, the word derived from the glyco means glucose and the lysis means breakdown. So, glycolysis is the common step of respiration, both in aerobic and anaerobic respiration. And it occurs in the cytoplasm of the cell, both in the prokaryotic and eukaryotic cell. So, glycolysis basically consists of the two phases. I will show you here this phase from the glucose to fructose 1,6 diphosphate or biphosphate. This is the preparatory phase, and from here, from this glyceraldehyde 3-phosphate up to pyruvic acid or pyruvate is the payoff phase, or you can say that the oxidative phase. Is it clear? So, glycolysis, which is the breakdown of the glucose, you can see here the glucose is the 6 carbon compound, and you can count number of carbon 1, 2, 3, 4, 5, 6. So this is the 6 carbon compound glucose, which is the monosaccharide, undergo the glycolysis in the cytoplasm, and this glucose was converted into glucose 6-phosphate. You can see here with the help of the enzyme hexose kinase. So this kinase enzyme help in the removal of the phosphate from the ATP and then attached to the glucose and from the glucose 6-phosphate. Actually, this is the preparatory phase. It is also known as the carbon activation phase. This phosphate attached with the glucose to activate the glucose. So phosphate come from the breakdown of the one ATP molecule at this step, and this ATP converted into ADP and phosphate released, and this phosphate attached with the carbon number 6. This is carbon number 1, 2, 3, 4, 5 and 6. This phosphate attached with the carbon number 6. Okay, now in the next step you can see here this glucose 6-phosphate is converted into fructose 6-phosphate with the help of the enzyme glucose isomerase, because glucose and fructose both are the isomers. So this step needs the enzyme isomerase. Is it clear? So glucose 6-phosphate converted into fructose 6-phosphate. So glucose and fructose both are the isomers, both are the monosaccharide, and you can count the number of carbons same because they are the isomers having same molecular formula but different functional groups. So glucose 6-phosphate converted into fructose 6-phosphate. Now, in the next step, you can see this glucose 6-phosphate is converted into this fructose 6-phosphate converted into fructose 1,6 diphosphate. You can also read it as a biphosphate. You can write it as a biphosphate or diphosphate. So fructose 1,6 or diphosphate. So this fructose 1,6 biphosphate produced as a result of the phosphate fructose kinase. Now this enzyme help in the again one molecule of ATP we need for this step, and this ATP breakdown into ADP, and now this phosphate come and attach with the carbon number 1 of the fructose. You can see here, so 1, and this is carbon number 6. So this is 1,6 diphosphate or biphosphate. Is it clear? Now I'm moving toward the next step and entering into the next phase. This is the payoff phase or oxidative phase. Now what will happen? This 1,6 biphosphate converted into 2 molecule. One is the dihydroxy acetone phosphate. This is three carbon compound. This is what? This is 3 carbon compound. Dihydroxy acetone phosphate. DHAP. You can pronounce it as DHAP, dihydroxy acetone phosphate. Now again, you can see on the other hand, it produces with the help of the aldolase enzyme, glyceraldehyde 3-phosphate, which is also known as G3P or PGL, glyceraldehyde 3-phosphate. Now, these are the two compounds which are 3 carbon. This is 6 carbon. Up to this phase, this is the 6 carbon compound. Now, these are the 3 carbon compound, dihydroxy acetone phosphate, and on the other hand, glyceraldehyde 3-phosphate. Now, this glyceraldehyde 3-phosphate enter into the next reaction with the help of the enzyme GLAP. We can use G3P or GLAP for an hydrogenase. Now what will happen? Here, this glyceraldehyde 3-phosphate converted into 1,3-diphosphoglyceric acid, or 1,3 you can also read it 1,3-diphosphoglycerate. This is also known as glycerate or 1,3-diphosphoglyceric acid. What will happen here? Now this aldehyde now converted into, this is acid, this is aldehyde. Now this aldehyde converted into acid. Is it clear? Glyceraldehyde, and this is phosphoric diphosphoglycerate or glyceric acid. Now at this step, what will happen? 2 hydrogen atom releases, and these hydrogen atom combine with the coenzyme 2-NAD. And this 2-NAD now converted into 2-NADH. Is it clear? Now what will happen in the next step? Now this 1,3-diphosphoglyceric acid is converted into 3-phosphoglyceric acid or 3-phosphoglycerate. You can call it as 3-phospho. You can read it as 3-phosphoglyceric glycerate or 3-phosphoglyceric acid. I am repeating again and again. Now this is 1,3 bi or di, and now this is 3. So here 1-phosphate releases. This 1-phosphate releases at position carbon number 1. This phosphate releases and combine with the or react with the 2-ADP. Here these are the 2 molecules. Must keep it in your mind. So here phosphate releases and combine with the 2-ADP and produces the 2-ATP. Is it clear? 2-ADP. Now 2-ATP converted into 2-ATP. Now the arrow is in the vertical position, but don't be confused about it. 2-ATP here produce 2-ATP. 2-ADP combined with 2-phosphate and produces the 2-ATP. The arrow is here wrong. Okay, now this is the mistake by Zainab. Okay, now in the next step, what will happen? This 3-phosphoglycerate or glyceric acid is converted into 2-phosphoglycerate. Now here there is an interconversion. Now from the carbon number 3, phosphate move towards the carbon number 2. Now this 2-phosphoglyceric acid or 2-phosphoglycerate is converted into phosphoenol pyruvate with the help of the enzyme enolase. Now phosphoenol pyruvate. Now what will happen? 2 water molecule releases from this step. Now at this step, what will happen? This 3-phosphoenol pyruvic acid or pyruvate is converted into 2-pyruvate or 2-pyruvic acid. But at this step, again 2-phosphate releases from the this phosphate releases from the phosphoenol pyruvic acid or pyruvate and combine with the 2-ADP. You can see here and produces the 2-ADP. And at the end, you can see the 2-pyruvic acid or 2-pyruvate produce. You can count 2 to 4 ATP molecule produce, 2 NADH molecule produce. So total, you can say 4 ATP molecule produce, 2 NADH molecule produce, 2 water molecule produce, as well as 2 proton produce. And out of the 4 ATP molecule which produced during the glycolysis used during the preparatory phase of the glycolysis, here you can see 1 and 2. So the total net gain of the glycolysis, the remaining amount of the ATP is 2 ATP molecule, 2 NADH, 2 water, and 2 proton. This is all about the glycolysis. I hope so you enjoy this lecture, and thank you, Zainab. The credit goes to Zainab. Actually, Zainab created the chart for my lectures. Thank you.